Hi to everyone. My name is Yoann Roupioz. I am a research director for the French National Research Center, and I'm working at the SIM lab based in France. So this is my pleasure to introduce this NanoAndes practical, which will be dedicated to the synthesis of a biosensor made of gold nanoparticles for the detection of adenosine. What is a biosensor? A biosensor is a device which is made of two parts. One part is usually functionalized with what we call a bioprobe, which is a molecule that will have some affinity toward a target that will be present in a sample. Indeed, if this target is present in the sample, then it will be captured by the bioprobe located on the surface of the device, and then the binding between the bioprobe and the target will induce somehow the production, actually the transduction of a signal that will be used for the detection of the target. Here are some details about all the different kinds of biosensors you may find around you or in the literature. Regarding the bioprobe, it can be a molecule which might be very different. It might be made of short DNA strands, it might be made of RNA, it might be made of PNA, which is a mixture between peptides and nucleic acids, it might be made of antibodies, enzymes, and so on. Regarding the transduction system, different strategies might be used for the production of the signal. It might be an optical transduction, which is the case in SPR-based biosensors, for instance, or fluorescence-based biosensors. It might be based on a shift due to the increase of mass on the device. This is the case for QCM, quartz microbalance system, which is based on piezoelectric device. It might be an electrochemical approach for the transduction of the signal. This is the case for glucometers, for instance. Maybe you know this device, which are used every day by people having diabetes. What's the difference between a microarray and a biosensor? In a biosensor, usually people are interested in the detection of one molecule or one target. In a microarray, actually you can run the detection of a large number of targets using a single device. So this is somehow a parallelized biosensor. Here are the main issues about biosensors and microarrays. When designing these kind of devices, you will always have to focus on the high specificity, high sensitivity, and ability to work in raw or complex media. What is a high specificity? The specificity will be the ability of your device to detect the right target, even though the sample contains other molecules or other targets whose structure and shape is very close to the one you are interested in. The sensitivity will be the minimum concentration you will be able to detect in the sample. So as you can guess, you will always try to design systems where the sensitivity is much lower than this concentration of the target you may find in your sample. The last point is the ability to work in raw or complex media. Indeed, most biosensors are made for working with biological samples, and usually these biological samples are very complex. It might be a blood drop, it might be some saliva, it might be urine, and so on. These samples may contain a large number of different proteins. It may contain cells or cellular debris and so on. And you have to make sure that the system is still working on this kind of solutions, even though there are many different ingredients in the sample. Here is one of the most common biosensor you may find in a shop. This is a pregnancy test. In the pregnancy test, you will detect a human hormone named human chorionic gonadotropin. So with this system here, you will drop a small volume of urine on the left-hand side here, and thanks to capillary forces, the sample will move this way and will pass over two lines made of antibodies. These guys here are very specific to the human chorionic gonadotropin hormone. And these guys here, which are labeled on particles, are also specific to this target. So if the target is present, everything will be moved this way, and then you will form this kind of sandwiches here, where the target is bound on the surface of the biosensor and also on the label. Okay, so in this kind of approach, it's an optical transduction. You would just have to look at the device to see if there is a positive response or not. And you will have something that will be very specific to the human chorionic gonadotropin thanks to the selectivity of these antibodies. Okay. The sensitivity of this kind of assay is usually about 17 milli-international units per milliliter. This unit here is a unit commonly used in pharmacology. Antibodies are very common and often used for biosensors because they are highly specific to epitopes located on the surface of an antigen. So as you can see here, one single antigen may display different kind of epitopes, meaning you can raise antibodies that will recognize different locations on the same antigen. I guess you all know that these guys have usually this shape. This is the case for immunoglobulins G, with two domains here, which are identical, and that will bind to the same epitope, and one fragment here, which is named constant fragment, that will be specific to a species, that will produce this kind of antibody. There are different ways for producing monoclonal or polyclonal antibodies. Antibodies are very common because they might be raised against different kind of targets, although it's very difficult to have antibodies specific to toxins or antibodies specific to small molecules or drugs. Antibody storage and stability is also an issue, and cost is very important for the production of antibodies. You can see here, this is a very basic and simple scheme describing the production of monoclonal antibodies. First, you have to immunize a mice or rabbit or horse with the antigen contained here in the syringe. Then the animal here will produce specific cells that are displaying antibodies specific to the antigen on the surface. Then these cells will be fused with tumor cells, and then these cells here, named hybridomas, will be selected, isolated, and cultured for the production of antibodies. This is a very long and expensive process. For this reason, since the 1990s, some people are developing other strategies based on nucleic acids rather than on proteins for the production of ligands that might be very specific to a target. These guys are named aptamers. So the objective is to mimic antibodies, but to take benefits of the nucleic acids, which are much easier to produce and easily chemically modified by introducing labels or fluorophores or grafting on magnetic beads, for instance. The production way to get these aptamers is named selects. Here, in the red circle here, you have the very first example of a DNA aptamer, which is specific to the thrombin protein, which is very important human protein. You can also raise aptamers against very small molecules, which is not possible with antibodies, as I said earlier. Here is the adenosine. So adenosine is a neurotransmitter. It's also a small natural compound which is involved in blood pressure control, and it's also a metabolic intermediate. Here you have the very simple structure of the first aptamer, which has been identified for the binding and specific binding of adenosine. So this sequence here has been published in 1995 and has been turned out to be very sensitive for the detection of adenosine. The KD has been measured and is in the range of 6 micromolar. Here are the crystal structures of the aptamer binding two adenosines. Actually, it's forming a pocket as shown here on this scheme, and in this pocket, two adenosine molecules can fit and be bound to the wall structure. So here you have the shape of the folded aptamer with the main pocket here, where two adenosine molecules are bound. So today, I'm very pleased to show you this practical for the NanoAndes network. So the objective of this practical will be the synthesis of gold nanoparticles, the coverage of these gold nanoparticles with oligonucleotides, which will be used as split aptamers, and then the use of these nanoparticles for biosensing the solution of adenosine, which has been set as a target in a biosensing assay. So first, we'll do the gold nanoparticle synthesis. You will see it's a quite simple step that will require only 3 reagents: water, gold salt, and sodium citrate. But before I get started, I just want to tell you that you have to wear lab coats, gloves, safety goggles, and work into a safety chemical fume hood. So the setup is quite simple. You have here a heating system. In the balloon here, there is 50 milliliters of water, of boiling water. And on the top of this flask, there is a cooling system. This cooling system is cooled by flowing fresh water into it. So the water comes from this pipe here, up through this system, and goes here into the sink. You have to make sure that here you properly tighten the flask here, but at this position here, it has to be loose to make sure that it can freely move in case of problem. Okay? So before we get started with the practical, I will show you how to use an automatic pipette and also how to prepare some reagents, like for instance, the sodium citrate solution. Okay, so now I will explain to you how to use an automatic pipette. Okay, the systems are made of 2 parts. This one is automatic pipettes, and then you have to use disposable plastic cones. These ones are single-use systems.
So you plug the plastic on this way, on the automatic pipette, and then you can adjust the volume you will need by turning the button here in this direction. Okay, if you turn this way, you will decrease the volume you can pipette, and if you turn the bottom the other way, you will increase the volume. You can see here through this window the volume which has been set. So here, let's say I need 50 microliters. Of course, I use an automatic pipette which is in the range of the volume I need. For instance, on this one, I can pipette up to 200 microliters. So usually, the systems are made to pipette volumes in the range of 1/10 of the value which is written here, up to the value which is written here. The plasticons which is used here is made for aqueous solutions. It means that if you want to pipette organic solvents, like ethanol or acetone, you may have leaks because this plastic is not made and suitable for the organic solvents. For pipetting, it's very easy. You just have to press this button. Actually, there are two levels. This one is the first level, which is the one which corresponds to the volume which has been set here. And then if you press further, you will have a second level which will be used to release the total volume of the liquids which is contained into the pipette cone. So for pipetting, you just have to first press till the first level outside of the liquid, then move the pipette cone just few milliliters below the level of the liquid, then gently release the button and fully release it, and then take the flask where you want to transfer the volume, move the pipette cone close to the wall of this flask, and then gently release till the first level, and before leaving, press fully the button till the second level. And then you're done. So now we'll prepare the citrate solution. So the citrate solution is made of this powder, which is sodium citrate, tribasic dehydrate salt. Okay, so we'll weight some of this powder to prepare a 100 milliliter solution at 1%. 1% meaning 1 gram of this powder per 100 of milliliter of water. So of course it's way too large. I mean, we'll not need 100 milliliter of powder. We'll only weight 50 milligrams that we will dissolve in 5 milliliter of water. Then it will make a 1% solution of citrates. $65 milligrams. Then I will adjust the volume to make sure that I will have the 1% solution, meaning that I have to dissolve these 65 milligrams into 6.5 milliliter of water. So everything is ready. I have my safety equipment. I can start the practical. So first, I will use this automatic pipette and pipette 50 microliters of gold solution. This solution is quite stable. It can be prepared in advance. So you can see that it has a bright yellow color. which is very characteristic to any gold salt solution. Then I remove the lid and add the gold solution into the flask. After that, I can use the citrate solution, which has been prepared just before the experiment. This is very important to make sure that you do not use an old solution and that you use a solution that has been only freshly prepared. So for that, I will quickly add 1.9 milliliter of citrate solution dissolved in water at 1%. 1% meaning 1 gram per 100 of milliliter of water. Now it's done. So you keep the heating system on, okay? And then you can observe the color shifts that will happen within the first few minutes after addition of the citrate. You can also start to record the time, making sure that the experiment lasts at least 30 minutes up to 1 hour. So the color is changing a little bit now. It's getting bluish or purple. And then it should turn as a deep red solution. And eventually it's turning red, which is a typical color of any gold colloidal solutions. So now it has been 30 minutes since we launched the reaction. So the reaction is completed. We can cool down the solution before using it. So to do that, you first switch off the heating system. You lower the wall system, and then don't forget to switch off the water flow through the refrigerating system. Well, so now we will record the visible absorbance of the colloidal solution. So as you have seen, it has a deep red color. So this is a colloidal solution which has been cooled down at almost room temperature. So first, I have to launch and define the conditions for the recording of the absorbance. So I use absorption spectrum, and then I have to set the minimum wavelength. So let's say 300 nanometers. And the pitch, let's say 2 nanometers. Maximum wavelength, 650. And that's it. And then I can launch the acquisition. I have a message that says that I have to first prepare the blank. So as the reaction has been carried in an aqueous solution, the blank can be recorded with deionized water. So this is what I have here. I have the cuvette here. So this one is compatible for small volumes. For instance, 500 microliters are sufficient for recording the spectrum. Then I can press OK, and it will scan the whole spectrum and use the references at the blank. So now the blank spectrum has been recorded. I can prepare the solution for the colloidal solution. So this is the gold nanoparticle solution. I can pipette 500 microliters once again. And then I can launch the acquisition for the sample. So here, let's say, these are gold nanoparticles. These gold nanoparticles are quite safe. There's no specific things to take care of, okay? Just make sure that you have the proper bin for recycling or disposal of the solutions. So the maximum wavelength is about 520 nanometers, which is consistent with gold nanoparticles in the range of 20 nanometers in diameter. Now it has been completed. So the absorbance at the maximum wavelength is about 0.98 absorbance units. So now the synthesis of the gold nanoparticles is completed. The gold nanoparticles are covered by citrate ligands, which is present on the surface of the nanoparticles. But before the coupling with the aptamer fragments, we have to displace the citrate molecules and to add phosphine, which will be way more convenient for the conjugation of the gold nanoparticles with the oligonucleotides, which contains the silyl moiety. Okay? So to do that, we have to add the phosphine, which is this reagent here, and we have to adjust the amount of phosphine depending on the concentration of the nanoparticle. Well, so we have to adjust the amount of phosphine to displace the citrate. Okay, so you remember that the absorbance of the gold nanoparticles at 220 nanometer was 0.98, okay? So it's about 1, okay? So this value here is very important. So it means that 1 milliliter of gold nanoparticle solution has 1 optical density units at 595 nanometers. So for each 1 unit here, we have to add 3 milligrams of phosphine, which is also named BSPP. Okay? We have 50 milliliters of this 1 optical density unit solution, meaning that we have to multiply 50 by 3 milligrams and then weight 150 milligrams of phosphine. Okay? So here is the phosphine. In this piece of paper, there are 150 milligrams of phosphine. And then I will add the reagents. Into the flask. Add the lid. And then I will maintain the stirring for at least 2 days at room temperature.